Let's dive into SDS page. It is a powerful tool used to separate proteins based solely on their molecular weight. But first, we have to prep the sample. We add a loading buffer containing SDS, beta-mercaptoethanol, glycerol, and bromophenol group.
And then, we heat the sample at 95 degrees Celsius for 5 minutes. Protein naturally exists in primary, secondary, tertiary, and quaternary structure held by hydrogen bond and hydrophobic interaction. This is because proteins are folded into a complex shape by hydrogen and disulfide bond. The SDS, which is a negatively charged detergent, is added to disrupt this non-covalent bond, causing the protein to unfold into a linear chain. Meanwhile, a beta-mercaptoethanol, a reducing agent, is added to break the strong covalent disulfide bond.
The SDS binds uniformly to the protein, coating it in a negative charge. This makes the protein's original charge negligible and turns it into a linear molecule. Once they are linear and negatively charged, they are ready to be sorted by size in the gel.
The gel used in SDS batch is a discontinuous system consisting of a top stacking gel and a bottom separating gel, which work together to first concentrate the protein and then sort them by size. The gel matrix itself is created through the polymerization of acrylamide monomers and bi-acrylamide cross-linker, which form a molecular sieve. To transform this chemical from a liquid into a solid gel, ammonium persulfate (APS) is added to provide the free radical necessary to initiate the reaction, while TMAT serves as catalyst to stabilize those radicals and accelerate the solidification process.
Don't forget the porosity. By changing the ratio of acrylamide, we control the pore size. Low concentration are better for high molecular weight samples. We also use a plastic comb to create wells where the sample will sit.
Once the gel is set, we place it in a chamber with a trisglycine chloride buffer. We also load a molecular weight marker, a set of known protein sizes, so that we have a reference. We load the samples into the wells, adding glycerol so the samples sink to the bottom rather than floating away.
Now we turn on the power. The negative electrode is at the top and the positive is at the bottom. Since our proteins are coated in negative SDS, they migrate towards the positive end.
Once they hit the separating gel, the race begins. Small proteins move easily through the mesh, while large proteins are slowed down.
This ensures all protein hit the separating gel at the exact same time so we don't get a smear result. In the stacking gel, proteins are concentrated into a tight, narrow band by being sandwiched between fast chlorine ion and slow glycine ion. This stacking occurs because of a lower pH and voltage gradient, ensuring all proteins enter the separating gel at the same time. Once they hit the separating gel, the glycine ions split up, releasing the protein to be sorted by size without leaving blurry or messy string.
We watch the bromophenol blue dye to know when to stop the run before the samples fall off the bottom.
Gel out and stain it, usually with chromacy glue. This binds to the protein so they appear as blue bands. By comparing our sample bands to the marker below the earlier, we can finally determine the molecular weight of our unknown protein.